like I said before, I'm planning a study and we're just underway with it on credit River Brook Trout. Um, I'll mention before I go to the next slide, the photo you're looking at was taken by Steve Noakes, who I think many of you know. His photography and also videography are on display throughout this presentation, and I'm thankful that he allowed me to use his stuff because the aesthetics of this whole thing would have been a lot less without it. So, yeah, as I mentioned, my name's Andrew. I'm a Ph.D. candidate in biology at Carleton. I'm a member of two labs there, the first of which is the Fish Ecology and Conservation Physiology lab. My focus is on the socio-ecology of freshwater fisheries, as well as conservation, social, and biological science. And one of my co-supervisors who's not here with us today is Stephen J. Cook, who some of you may also be familiar with. Steve's the founder and the primary investigator at the lab that I just mentioned. He's a professor at Carleton. He has a number of different affiliations, and his research interests and topics are very broad and deep. Steve's also a former Canada Research Chair, a Royal Canadian Society Fellow, and a Royal Canadian Geographical Society Fellow. So I'll talk a little bit about the Cook Lab. I'm just scrolling. If you can see the video right here, this gives you a sense of our lab website, which you can visit, and some of the stuff we're doing. So to give you a little bit of background on the study species, not sure that it's entirely necessary for all of you, but brook trout, or as they're sometimes known in the scientific community, Salvelinus fontinalis, are actually a fish from the char genus in the salmon family. They're native to the northeastern United States as well as eastern Canada. And there's also a variety of non-native populations in the western US and Canada. So brook trout sort of, they get around. But our interest is in brook trout that are native to eastern Canada and the United States. And the project really revolves around the idea that, well, the hypothesis, I guess, is more correct to say, that brook trout are sort of on the edge. So at areas to sort of the southern end of their native species range, which includes our region, I think brook trout are threatened for a variety of reasons. And you can see I've included a map in the top right here of the native range of brook trout, which you can see, we're in our region and the Credit River and surrounding area, sort of at the southern end of that. And the areas that have wild brook trout south of there are mainly at elevation. They exist just because of the Appalachian Range. So brook trout, it's important to note, are what we call a stenothermal species, which is a lot different than a euthermal species like a channel catfish, which is why I've included a picture there. And the difference between the two is stenothermal species can only tolerate a very narrow range of temperatures, whereas eutherms can tolerate quite a wide range. And this is a problem given climate impacts that we're already seeing and that we expect to continue into the future. And brook trout are also impacted by what's now becoming recognized widely as the freshwater biodiversity crisis. So freshwater biodiversity compared to marine and terrestrial biodiversity has declined more and is declining more sharply. And brook trout are included in this. And that's the result of not just climate impacts, but local impacts, which I'll talk a little bit more about in the next. So a little bit more background in this case on the Credit River. The Credit River, the upper Credit River that is, has a really healthy population of brook trout relative to the surrounding region. This is mostly from the cataract upstream to Orangeville, Erin, and beyond. So these fish are healthy and abundant compared to a lot of populations in the surrounding region. But they face threats of their own, and they provide a great opportunity for us to study brook trout. So a big reason why we're doing this sort of research is because of emergent and intensifying anthropogenic stressors, both at a local scale and at a global scale. So locally, things like urbanization, agriculture, fragmentation, exploitation, these are impacts that impact all fish species, but they're particularly potent when it comes to brook trout and sort of sensitive stream environments and cold water habitat like this. And then global scale threats like climate change and aquatic invasive species add to that. So Between all of that, brook trout resilience in the credit and beyond is really going to be tested going forward. It already is, but as I mentioned, these things are expected to intensify and other threats are going to emerge. And so I think we need to understand more about brook trout in order to protect them, given what might happen in the near future. So a little bit more, well, I'll talk now about the project specifically. So as I mentioned before, a lot of this revolves around the idea and the hypothesis that brook trout are sort of up against it. So I think that populations at or near the southern fringe of their species range are going to be at risk of extirpation due to the cumulative effects that I've already mentioned. So I think brook trout are going to rely increasingly on, habitat that's more and more scarce and isolated in light of the environmental changes that are anticipated. And I think anthropogenic stressors are going to force brook trout to adapt behaviorally as well as physiologically and genetically to novel conditions that they're going to see. So I've got a few main objectives here. The first is to better understand and determine what the habitat requirements of brook trout in an environment like the Credit River and in the surrounding region are, and how they use that habitat. I want to understand more about the health of those brook trout and how healthy they are in relation to anthropogenic stressors and under stress. And by doing that, I'm hoping to come away with some guidelines, some recommendations, broadly speaking, directions for evidence-based management and conservation. So I've got three main questions that I want to answer here. The first of those is, how does the use of stream habitat by brook trout in Southern Ontario change seasonally as well as in response to anthropogenic stress? And what are the critical habitat components that support brook trout in the region? Second question is, what anthropogenic stressors affect brook trout in a typical Southern Ontario stream and to what degree? And the third and final question I want to answer is, how can brook trout populations be managed and conserved in light of all this? So I think I can answer these questions with two basic components of my study. The first one is, relates to spatial ecology. So spatial ecology, roughly speaking, means spatial patterns in nature. In the context of brook trout in the upper credit, this means the use of habitat by those fish, their range and patterns of movement, subpopulation structure, and other things like that. And what's of most interest to me is habitat components and things like tributary streams, groundwater, physical barriers, things that affect the brook trout's ability to use their habitat and to survive and thrive in that habitat. So one of the things I've started doing, and this is the very first stage of our actual on-ground work, is stream walks along the credit. We had one last Saturday, and there will be many more before we get started on, some of the aspects of our project that'll begin next month. But yeah, this stage, we're basically identifying these points of interest and the different habitat components, and this will allow us to study the behavior of brook trout. And that's a correlative thing. It'll allow us to infer some things about the health of these populations, but there are ways to study that more directly too that we also plan to use, and I'll talk more about that in a minute. So acoustic telemetry, I'll just briefly describe this. This is the method that we're going to use to study the spatial ecology of brook trout, and it's really fascinating. It's a good example of a fairly novel technology and method that gives us fairly novel insights on brook trout and allows us to answer questions that were a lot more difficult to answer in the past, like where do brook trout go? What do they do at different times? There are ways to study this and infer things from presence or absence, electrofishing, even angling. Anglers, I think, tend to have a lot of great knowledge on this, but I think acoustic telemetry provides us a great opportunity to study this in more detail. So I've included some pictures. The little object you see next to the paperclip is actually an acoustic transmitter. So what we do typically is we implant these in a fish surgically, and then we install throughout the river the object that you see to the right, which is an acoustic receiver, and that's about the size of a wine bottle. And we can place these throughout the river at regular intervals. And when a fish passes by it, the acoustic transmitter emits a signal, a sound, and the acoustic receiver picks that up. and then we can tell definitively, as long as we can rule out a false detection, that that fish was in that place at that exact time. And so by installing a number of these receivers up and down the river, we set up what we call an acoustic array. And then we can watch how these fish move around the river across seasons. And, you know, we can determine things like how are they using their habitat? What areas of the stream do they need to survive at different times in the season? And this is distinct from radio telemetry, which I'm not sure if anyone's heard of that before, but what's really nice about acoustic telemetry is we put the receivers in, we implant the tags, and then as long as they're in there, they're picking up signals and they're monitoring the fish movement. And then we can download data at the end of the season and, you know, get really detailed info on how these fish are moving and using habitat. So the second component, which I briefly alluded to, is physiology. So I'm going to study brook trout physiology, and this relates, you know, directly to the function of these animals at the level of their cells and organs, etc. Something important to note about brook trout, but also most freshwater fish, is that they're ectotherms, which means they don't thermoregulate physiologically. They're not like us where they can sweat or cool down in, you know, the ways that we or other mammals or animals can. They essentially take on the temperature of their surrounding environment. And this is a big problem when we see impacts from climate change, for example, because brook trout don't have the ability to adapt to this the way we do. Brook trout can acclimate like a lot of other freshwater fish can, but that ability is fairly limited. They have a preferred temperature, obviously. I think everybody probably knows that. Roughly speaking, it's something like 14 to 18 degrees Celsius. And within that zone, brook trout will sort of optimize different performance attributes, you know, metabolism. Brook trout will avoid temperatures equal to or above 20 degrees Celsius, and beyond 24 degrees Celsius, brook trout start to die off in a major way. I mentioned that they can acclimate, but that potential is limited. There are some differences across brook trout strains in the ability to acclimate, but once you get up to that zone of avoidance and that you know, lethal limit of around 24 degrees Celsius, those differences sort of fade. And, you know, once waters get that warm, brook trout just aren't going to do very well. So conservation genomics. Basically, this means applying genomic analysis to biodiversity conservation, looking at things like population structures and divisions, genetic diversity, the health of populations and how populations are adapting. And this is, like acoustic telemetry, a great example of a fairly new method and technology that provides fairly new and much more detailed insights about brook trout. We can look at things like cumulative stress, and cumulative stress is a concept that I think will be familiar to most of you on this call right now, because a lot of you are anglers and you're, you know, have probably abstained from fishing at times when the water temperature was particularly high. And this is because, you know, of cumulative stress, right? Thermal stress plus angling stress. The impact of that is going to be greater than the sum of its parts, right? So genomics give us a great opportunity to study that in detail, and we can look at genes and know much more definitively and in a more detailed way what's going on with brook trout. So this is facilitated by GenFish, which I'll talk briefly about. GenFish stands for the Genomic Network for Fish Identification, Stress, and Health, which is a research team and initiative funded by Genome Canada, of which this project and I am a part. One of GenFish's projects is called the Fish Health Toolkit, and that's something that I'll use in the Credit River. It's super useful. We can do things like determine what stressors are present in a stream like the Credit. How are the brook trout in the Credit River adapting to those stressors? And, you know, how healthy are those fish overall? So this is a study method in the field of epigenetics, studying environmentally and behaviorally mediated changes in gene expression. So not changes in the genome of an organism exactly, but changes in how those are expressed. So we've settled on two main study sites. The first one, which I'll call Site 1, is the Credit River main stem from roughly Highway 24 to Shaw's Creek, Alton, that area. I think this is a great study site for a number of reasons. For one thing, it provides us an opportunity to study thermal impacts just because water does get fairly warm on certain parts of this stretch. There's also good connectivity here to compare to other parts of the Credit River and brook trout streams in the surrounding region. So brook trout can really move up and down this stretch unimpeded, and it gives us a chance to see, for example, how far are these fish moving? Because they can move up and down quite a bit. This is a stretch of river where our plan is to implant 30 fish with acoustic tags and then install something like 15 acoustic receivers up and down and look at how these fish move around. The west credits are second site, roughly speaking, from Bell Fountain to Erin. Although, we'll get more specific about this once we've walked the stream a little bit more and planned a little bit more. We're really glad partnering with some of the individuals, particularly in Trout Unlimited Canada, has allowed us to, access this site. There's a ton of interest in this site by them and other partners. The West Credit's a special place. There's been a lot of concern recently about development and wastewater treatment plants in the town of Erin. And so, this makes it a great candidate for our study too, because if we can go in there and clarify what's going on. I think it's important to note that there's a lot of great work ongoing in the credit, mainly by NGOs, clubs, and conservation authorities like CVC, Trout Unlimited Canada, and of course the Isaac Walton Fly Fishing Club. So my hope is that I can add to the exceptional work that's already going on in the credit. I think groups like yours, groups like Trout Unlimited Canada, conservation authorities, in this case the CVC, make critical contributions in this area. And that includes things like monitoring, restoration work, advocacy, status reports, watershed studies, and even scientific research. So that's the second part of this slide. There is academic research already ongoing. There's been brook trout research, research on brook trout ecology and physiology, as well as research specific to the credit. I think most notably, Zimmer et al. In 2010 studied brown trout and the credit a little bit lower down than I'm planning to go in, but they looked at that was brown trout seasonal movements and determined that those were sort of driven by temperature and life history. They noticed that brown trout were fairly sedentary in between seasonal transitions. And, another interesting finding was that flow, volume, and turbidity had a fairly negligible effect on brown trout in the lower river. But it remains to be seen whether or not this is true for brook trout in the upper Credit River. And that's a question that I'd like to answer. So, are the effects of fragmentation and flow and turbidity greater for smaller fish like those upper Credit River brook trout? There's been some other interesting research too on, the impacts of wastewater treatment plants on Credit River brook trout, as well as, the use of stream habitat during winter by Credit River brook trout, as well as brown trout spawning, as well as the feeding and distribution of crustaceans in the Credit River. So a lot of interesting research and really, really high level work going on. And my hope is just that I can add to that. So this is my approximate timeline for the project. We had to push back the first part of that a little bit. So the tagging and receiver deployment that I originally wanted to do in April, it looks like now will probably happen in May. The cold treatments is what I'm calling the first phase of our genomic study, where we'll go in and look at the health of brook trout and the impacts of something like angling under relatively cool conditions, or sort of in that optimal temperature range for brook trout. And then later on, probably around July, but it'll depend on how our season shapes up, how the weather is, I'll go in and do the warm treatments. So I'll look at how brook trout are doing when the water's fairly warm, and we expect them to be stressed at that point, and we'll study the impacts of angling, for example, on brook trout under those same conditions. And then in between all of that, we'll be doing a fair amount of monitoring. So going in and checking that our acoustic receivers and our array are doing okay. We'll be monitoring temperature, dissolved oxygen, things like that. And then if all goes well, at the end of this summer, I'll go in and I'll download data from our acoustic receivers and take a look at how brook trout moved around and use stream habitat during the summer 2022 season. Our acoustic tags have a life of about 465 days on the settings that we've programmed them with. So I'll go in again in a little over a year after we've implanted those tags and download data once again. And at that point, I'll have a picture of brook trout spatial ecology in the Credit River over the course of an entire season. I'll briefly mention my appreciation for local organizations and the support from project partners that we've already received. I've already talked a little bit about some of the groups operating in the credit, but GenFish I've also mentioned. One of the really nice things about GenFish is it connects this project to government and the individuals that are responsible for managing our fisheries, including Ontario Ministry of Natural Resources and Forestry. I know they're not known by that same title anymore, but I haven't gotten the acronym down, the new one. As well as Fisheries and Oceans Canada. So we're connected to higher levels of government through the GenFish network, and that's really good. I think that keeps us integrated, and it ups our chances of making really a meaningful contribution. There'll be opportunities for individuals like yourselves to support and contribute if you're interested. Part of that will be field work. So there may be opportunities to help with stream walks, for example, to provide input, to provide your local knowledge on the credit, which will help inform what we do, and potentially to come help out catching fish at some point. I'm hoping there will be opportunities for that, but I'll keep everybody posted on those things. Another area where we're seeking contributions, and we've gotten some already, but just, if anyone's interested in contributing this way, we'd appreciate it, which is with equipment, which, can be a bit pricey. Our acoustic transmitters cost something like $200 a unit, and the batteries, which we need about 3 a year for, are around 100 a unit. So we've gotten some help paying for these so far. We're welcoming additional contributions, but I think we're going to be all right. So whatever support we can get is welcome. Otherwise, the project is in a pretty good place right now. The lab that I mentioned previously has already funded this project with quite a bit of money. Things like researcher salaries are covered. So like I said, we're just looking for small additional contributions when it comes to equipment. And then, my biggest hope is that we can give back to you a little bit. If there's anything your group specifically or individuals within your group are wondering about Credit River Brook Trout, let us know. I mean, if as part of our research, we can answer some of those questions or, help you resolve some of your concerns, for example, about Aaron's proposed wastewater treatment plant, we'd love to be able to do that. So definitely let us know. So you folks are anglers and stewards of the Credit River. I know you have local knowledge about the system, and you may have questions of your own about our project or the Credit River. At this point, I want to open the floor up, and I'd love to hear those things.
I know that in some parts of the areas around Haliburton and Algonquin Park, you can see some rivers and lakes have wild brook trout, at least listed in the booklets that I look at, and then they'll have stocked brook trout. So is there interaction between the two? How does that affect genetics? Is that something to think about?
Yeah, for sure. I think generally speaking, stocking works good as a last resort. I think stocking on top of a wild population, it's generally not the best thing. It was hard to know in the past, I think, because, without genetic technology and techniques, you can't really know exactly what's in a gene or what's in a specific brook trout population, but they tend to be really specifically evolved and fine-tuned to the environment that they've evolved in. And yeah, when you lose that, it's just so difficult to recover. So where those wild fish exist, I think it's really important to keep them wild. They're just more adapted to the environment that's there, right?
On the credit, in the warm summer periods, they always have signs up related to not fishing above a certain air temperature. And I've always wondered about, is that set too high? And what's the relevance of the air temperature to the water temperature? And that people should really be tracking water temperature, not. But I guess you have to give the public some guideline above which not to fish. But have you had any data supporting the air temperature that's currently set? Is it a reasonable level, or should it be set lower? What are your thoughts?
Yeah, I think the air temperature, as I understand it, those signs, it's so people can follow it, I think, easily. I think you know, TU thinks, I think, correctly, that the average person, although I think a lot of individuals in this group probably do, a lot of the people going to fish in the upper credit aren't going to carry a thermometer and monitor water temperature as diligently as they might need to. And so that sort of, I think it's 28 degrees Celsius, is set up as sort of a precautionary thing. If it gets to that point, asking people to not fish sort of is a catch-all, I think. But yeah, you're right. It all has to do with water temperature and also dissolved oxygen, which isn't necessarily linearly related to water temperature, although they are related. And yeah, it's more of a
I guess,
thing, I
yeah.
guess.
I guess my concern is 28 feels high as a it feels like once you're at that 28 level, you've probably already passed the water temperature at which you should be fishing. Should we be fighting to have that lowered? I know there's a lot of fights. You have to
Yeah.
pick your battles. But what are you? Yeah,
Yeah.
is
Well,
that too high a temperature? I guess is my question.
yeah. Well, it could be. I would, before saying anything definitively about that, I'd like to ask someone at TU their reasoning behind it. It's funny. It seems like, every spring now we're getting like heat waves in May. So, if we get that first heat wave in May and that sort of, we hit 28 at that point, the water's probably still quite cold then. But yeah, once we're, if you're hitting 28, 30 degrees in July and June, yeah, the water temperature is probably getting pretty high at that point, I would think. And one of the sort of, auxiliary questions I have is, would a heat closure be something that might be wise to do on the upper Credit River? If we find that when the water temperatures get towards 19 or 20 degrees, those fish are severely stressed and, responsible catch and release angling isn't likely. That's something that I might, recommend in, my publication or whatever comes out of this, is maybe we need to start thinking about a heat closure and, monitoring water temperature. And once it hits this point, no fishing, because that's something that we don't have any of that in Ontario. There's places out west where they have that, but it's something that we are going to need to think about, I think, because things are changing pretty quickly. And like you said, the water gets hotter and hotter earlier and earlier in the season and for longer periods of time. So I think, yeah.
Yeah, and to that point, actually, I think that's a great thought of having a closure, let's say midsummer, or specifically, measure the temperature and close the river when the temperature is too high. But our seasons have always been set to be closure in the fall, fairly early in the fall, way before spawning. But then it's not open in the spring when the temperatures are safe for the fish, and yet we're open in August when it's likely not safe for the fish. And so, hopefully your research can help guide some different regulations that are more in line with maintaining the wild populations we have without stressing them.
Yeah.
Dean, could I ask you, Dean, would you like to just add a little bit about what you found, the correlation between air temperature and water temperature? And
well, mostly we've looked right now at the sewage effluent, right? But like what we found with the sewage effluent is, so the coalition put loggers in 5 Southern Ontario wastewater treatment plants last summer. And we found some interesting things. There's pretty much a 1-to-1 correlation between the effluent temperature and the air temperature. So this is what's coming out of the sewage treatment plants, is that when the air goes up, the effluent goes up. When the air goes down, the effluent goes down. And then on top of that, there's like a background increase in the effluent temperature. So the effluent temperature, like a lake or a body of water, starts off around the same temperature of the river in June and July, and then accumulates heat. And then by the time you get to August, The effluent temperature is significantly hotter than most cold water rivers. And this continues into September and October. It takes a long time for the effluent to cool down. Right. And so the other thing that was done was there were loggers put upstream in the effluent and downstream of 3 sewage treatment plants. I believe it was Georgetown, Orangeville, and Acton. And the most interesting one is Acton because it shows Orangeville, the effluent is actually colder than what's coming out of Island Lake. So Island Lake gets quite hot in the summer. And then the effluent that's coming out of the Orangeville wastewater treatment plant is actually colder than what's coming out of Island Lake. So the effluent cools the river. But in the Acton, which is in the headwaters of Black Creek, the effluent, like it's by the time you get to August, the upstream versus downstream. So this is the creek upstream of the effluent of the wastewater treatment plant versus downstream. You get about on a daily average, about a 0.5 degree Celsius higher downstream than you do upstream. And then by the time you get to September, it's about 1 to 1.5 degrees Celsius hotter downstream than upstream. So like pretty significant change in the river temperature and the river. You even see it in the change in the diurnal variation. The downstream is like dampened, hotter and dampened. And so like, temperature was something that was never really on anybody's radar for the Aaron plant. And I think it was a mistake. It was kind of like, temperature had never really been considered. No effluent in any municipality had ever been cooled before. And the minister just said, "Oh, it's fine. We'll just go ahead with it." And we think that's a big mistake.
Yeah. That's great. Yeah, thanks,
yeah.
Dean.
Yeah, and like Randy, people from our club went out and stuck loggers in and donated loggers and stuff. Like there was probably 12 loggers deployed between May 1st and the middle of September. Like between Trout Unlimited, Canada, us, an organization called Ontario Streams, the OFAH, Ontario Federation of Anglers and Hunters, did a bunch of the work too. It's pretty impressive. So we'll see. And we're using that information to lobby the Ministry of Environment, Conservation and Parks to get cooling into the Aaron plant. So even when you do the fish surgery, do some of the fish die after the surgery? Like only live like a day or two after the surgery?
It's a risk. If you do the surgery well, no. There's a lot of things going on. There's the risk that they'll die. It's always a trade-off. I mean, if a couple fish die, in order for us to know more about them and help protect them, that's the trade-off we're constantly working with. But we've got a number of ways to sort of minimize that risk, and my aim is for 0 mortality. Basically, I can describe the surgery process a little bit and sort of what affects survival.
But another thing is, have they done it with like brook trout or rainbow trout or bass or something, and most of them survived?
Yeah, so I've done this sort of work with actually hatchery steelhead at one point. They're stunted hatchery steelhead, so a lot more like a rainbow or a resident trout in terms of size. We had 0 mortality in that case. The other, I guess, probably most comparable thing would be with juvenile salmon, which would be a little bit smaller than these trout that we'll be working with. And again, you don't get passed for a collection permit unless you've got some evidence to suggest that it's going to be fairly minimal in terms of stress and trauma on the fish. One of the things that could affect that is how they're captured. If they're captured in sort of a compromised state, whether that's, they're bruised from electrofishing or, they've been injured recently and we can tell that, those fish should be excluded from the study and just allowed to recover. We don't want to work on any fish that are compromised previously. But to give you some idea of the surgical process, basically, we would catch fish and sort of have holding pens prepared and, have all the surgery stuff set up in advance so the process is quick. Basically, electrofish the brook trout, net them, hold no more than probably 3 fish at a time in one of the holding pens, and those would be in stream with water flowing through them. So we'd monitor them in there, make sure that they're doing okay, and then we would take them out and we actually handle them with electric gloves, which sounds harsh, but it's not. basically, if anyone's done physiotherapy where they use a TENS unit to sort of stimulate muscle contraction, we use the same sort of unit in these gloves that electric current flows through. There's an anode on each glove, and when you hold a fish with that, you close the circuit and you basically immobilize the fish without having to use a chemical sedative like clove oil. And the advantage of that is as soon as you release them from those gloves, they recover instantly. So you hold them like that, and basically you make a small incision, probably about a centimeter long, insert the tag, and then put 2 sutures in, and then the fish goes back into another holding container where we monitor them, make sure that they recover and that they're writing properly, and then release them back into the river. So that's the process. There's obviously risk involved. There's obviously a certain amount of stress involved. Another thing that we are gunning for is to do it in cold water rather than doing it later in the season, right, where, but that's essentially what the process looks like. Yeah.
Yeah. But basically they've done it on other fish and it worked out.
Yeah. It's very common. If you look up acoustic telemetry papers, you'll get a whole bunch of different examples. Yeah.
Okay. And then if a fish does die, can you guys find the transmitter? Like, will you try to figure out where it last pinged, or will you take a portable whatever and go try to find it?
Yeah, we could definitely look for a last ping, but that'd be a little bit difficult, just because, we'd have to download data, and that's a lengthy process. I'm actually looking into whether or not there's a portable unit that we could go downriver and, look for pings, but
another one of the highly likely you send out 38 transmitters and you get back like, whatever, 23 or something, 25 or somewhere in there. Anglers keep fish, but aren't supposed to, but they do, right? The fish swim up to Aaron where you can keep them, and then that's it, right? The transmitter's gone.
Exactly. It's a constant risk for sure. That's why we do 30. So, hopefully you get a good sample. If a couple are eaten by herons, if a couple are eaten by fishermen, we've still got somewhat of a sample out there. Right.
Okay, then my final question here. So for the you said for the expressing of genes, so you're measuring proteins in the water? You're measuring like the proteins expressed by the genes, or you're measuring the actual some kind of DNA stuff in the water?
Right. So I guess I should have clarified that a bit. Basically, we take a non-lethal tissue sample from the fish.
Okay.
That sample is a gill filament, one of the red filaments in the gills. You take one of those.
Okay.
It sounds kind of funny to an angler who, wants to avoid any contact between hook and gill, but with a sharp scissor, taking one of those has little to no impact on the fish physically. And so you take that, and what we're looking at is what's called transcriptomics. So the transcriptome is basically I don't want to talk too much shop here, but it's the set of all the RNA transcripts. Basically certain genes, associated with immune function or, other physiological components.
So I'm talking like grade 10 biology, like DNA goes to RNA goes to protein. That's like, so you're measuring RNA, the RNA in the cell, in the tissue sample.
Yeah. What we're looking at is basically up regulations or down regulations of certain genes. So under different environmental conditions and, in response to stress, certain genes will be expressed differently. They will either upregulate or downregulate. And then, if that's happening, if we can identify a certain upregulation or downregulation, it's previously been associated with a certain stressor in the lab. Then just by looking at that, we can tell, okay, this was identified, this was associated with aquatic invasive species stress in the lab. And by taking a sample from a Carleton River Brook Trout, we can see, is this fish subject to that particular stressor, or is it experiencing thermal stress? And, we can look at a variety of things. We can look at, like I said, those genes associated with immune function and tell, under these conditions, how healthy are these fish? What are they experiencing? And,
so you have tissue samples from a lab where you stressed fish for temperatures, let's say, and then you're measuring something, the amount of RNA or the amount of whatever it is, and then you're measuring that also in the fish in the river. You're saying if the fish in the river pattern matches the fish in the lab, these guys are temperature stressed. That's the idea.
That's essentially it. By ground proofing that tool, like, our hope is that tool is going to be available and used more widely. So you go out and, monitor Carleton River Brook Trout, for example, and a lot of the time how healthy those fish are is kind of a mystery until they get really unhealthy. And the hope with this is that, you know, we'll have a tool that is relatively easy to use and go in and get a really detailed picture of how healthy are credit river brook trout, you know, without having to go and do a ton of research.
Just one quick question, if it's a good time. With respect to competition between, thermal competition between brown trout and brook trout, is that possibly a consideration in the, I mean, obviously your study hasn't started, but do you think that that could be a factor? For example, I imagine there'll be specific areas where springs, etc., are identified and other salmonids might all congregate there. Is there any previous studies that you're aware of that have shown competition between brown trout and brook trout in the past? And if that's something that may be relevant, and I'm just muting here your answer.
Sure. Yeah, I'm not aware of a specific study. That there may very well be studies out there that look at exactly that. I know brown trout tend to be a little bit more hardy when it comes to temperature. Like they can tolerate sort of a wider range, and they seem to do a little bit better at higher water temperatures than I think any other, you know, at least brook and rainbow trout. That won't so much be a problem on the Highway 24 stretch, just as there aren't brown trout up there. I know the West Credit does have the odd one in there, but I would expect there to definitely be some impact, just because, like you said, when the river warms enough, those fish are sort of heading to thermal refuge areas, and those can be pretty limited, right? So competition for space and food and things like that would presumably be much more intense in those spots. I think there's a chance that maybe we'll see that with just within brook trout populations on the upper river, right? Like the more limited the habitat is at sort of the height of summer or even the height of winter, I think, you know, the smaller that bottleneck is for the whole population, right? So I think that's a great hypothesis. And, you know, whether or not I can sort of answer that question, I think it deserves to be answered. It's a really good one.
It sounds like the sort of information that you're collecting is very disparate, like once every 3 months or once every 6 months. But do you consider environmental conditions and fish behavior as part of the study right now, or is it purely a health and water related study? And what I mean by that is, just to clarify, today is rainy, but it's 90% humidity, fish are hiding or fish are moving. That sort of aspect of the study, is that in consideration or not really?
Right. So do you mean sort of movements on the scale of days and weeks, you mean?
Days,
Yeah.
weeks, hours, that sort of thing. Yeah.
Right. So yeah, the plan is to go in and download the data at the end of the summer, but those receivers, the beauty of them is they're collecting data as long as they're in the stream. Yeah. So if those fish are moving in and out of an area, you know, just on the scale of hours, we're going to be able to detect that change. You know, the approach to analyzing it presumably will be to go and correlate it with things like temperature, you know, any other data that we have for the credit river. But yeah, like as long as those receivers and tags are in there working, we're recording fish data, and the settings on the tags are, you know, they ping every 15 seconds. Okay. So, you know, that's the scale we're working on basically, and those receivers are working that entire time. So I'm not sure if that answers your question.
It does. What I was really trying to get at was, is there equivalent logger basically installed on land to capture the two timestamps together to correlate the data down to the minute or to the hour as to what the fish's behavior is on that day versus the environmental conditions around?
Johan, I think I might be able to add a little bit of light to this because part of one of the grants that we've applied for, we're hoping that we'll be able to set up a constant visual monitoring of one area of the stream. So it'll be like a fish ladder, you know, where you have, so that might give us some clues as well. It'll be rudimentary at the beginning, but we're hoping that we can expand on them.
No, by all means, I was just wondering if it's part of the study at this stage, or is that a whole separate sort of subdivision as to fish's behavior versus their health overall?
Yeah, I think the two components, the telemetry component, the spatial ecology stuff, I think that sort of that covers the behavior, at least for me. And then the when it comes to their actual health, the genomic stuff, that's where that really comes in, because we can really definitively tell how healthy they are by looking at their genes.
Yeah. Okay. Thank you.
I just want to add two things. One thing, I did a lot of electroshocking up in the Highway 24 section, and a lot of the brook trout have a black dot disease, which is a parasite because of the high water temperatures. The other thing too, on the other stretch that we're studying, just upstream of Bell Fountain, there's a couple of bridges up there, and just downstream of one, I remember one time, I guess it was a warm summer day, I saw probably 100 brook trout just crowded around this one little area where there must have been a spring.
Yeah, I know there's a number of those on the Upper Credit. And like you said, I think they become obvious in summer when all those fish have to flock to them. I've been working and going back and forth quite a bit with Phil Bird, John Clayton, Steve Noakes, folks like that who, really have their thumb on the pulse of Credit River Brook Trout. And they've kindly pointed out some of those areas to me. So that's a great example of one of the habitat components and features that I'm really interested in. So if I can identify those, I can position the receivers accordingly and try and see, I guess, document how those fish are moving and using that habitat across seasons. You also mentioned the black spot disease, the parasite, and that's a great example of, I talked earlier about temperatures and the relation of temperatures to performance attributes in brook trout. And one of those performance attributes would be immune function. So yeah, once you get water temperatures that are sort of out of that optimal range, one of the things that we expect to decline is immune function. And so, yeah, it makes total sense that warmer water temperatures, and when those fish are more stressed, they're also much more vulnerable to parasites. So, and thanks again, everyone, for not just, for having me. Thanks for the comments. Thanks for the questions. This was a pleasure for me, and I hope it was informative.